Welcome to my channel XY Biology Academy. In natural environments, microorganisms rarely occur as single species. Instead, they exist as mixed populations containing numerous types of microorganisms. For the accurate study of an organism's cultural, morphological, physiological, and biochemical characteristics, it must first be isolated in the form of a pure culture. The streak plate method is one of the most widely used microbiological techniques for obtaining pure cultures. It is a mechanical dilution technique in which a small amount of mixed culture is progressively spread over the surface of a solid agar medium. Sterilization of the inoculating loop between successive streaks progressively reduces the number of bacterial cells transferred to each new section of the plate. This gradual dilution separates individual bacterial cells from one another across the agar surface. Materials required: mixed bacterial culture, culture media, sterile inoculating loop, Bunsen burner or microincinerator, marker or laboratory marking pencil, test tube rack, incubator. Procedure. Step 1. Labeling the plate. Label the bottom surface of the sterile agar plate with the experiment title, date, sample identification, and initials. Always label the base of the Petri plate rather than the lid to prevent misidentification if the lid is accidentally removed. Step 2. Sterilization of the inoculating loop. Sterilize the inoculating loop by heating it in the flame of a Bunsen burner or microincinerator until the wire becomes red hot. Allow the loop to cool by gently touching an unused sterile portion of the agar near the edge of the plate. A hot loop should never be used to collect bacterial culture, as it may destroy the microorganisms. Step 3. Collection of the inoculum. Flame the mouth of the culture tube, insert the cooled sterile loop into the broth culture, and collect a loopful of the mixed bacterial suspension. Flame the mouth of the tube once again before replacing the cap. Step 4. Streaking the first quadrant. Partially lift the lid of the Petri plate and streak the inoculum over area 1 using closely spaced parallel streaks. This first quadrant contains the highest concentration of bacterial cells and serves as the starting point for subsequent dilution. Step 5. Sterilization before the second streak. Sterilize the inoculating loop until it becomes red hot, allow it to cool, and rotate the Petri plate approximately 90 degrees. Step 6. Streaking the second quadrant. Touch the cooled sterile loop once at the edge of area 1 and drag the bacteria into area 2 using several parallel streaks. Once the streaking of area 2 has begun, do not return the loop to area 1. Step 7. Sterilization before the third streak. Sterilize and cool the inoculating loop again. Rotate the Petri plate another 90 degrees. Step 8. Streaking the third quadrant. Touch the edge of area 2 once and streak the bacteria throughout area 3. At this stage, fewer bacterial cells are transferred, resulting in a greater degree of dilution. Step 9. Streaking the fourth quadrant. Rotate the Petri plate another 90 degrees. Following the standard 4-quadrant streak method, drag the bacteria from one corner of area 3 into area 4 using a wider streaking pattern. Avoid touching any previously streaked areas except at the initial point of contact. This final streak provides the greatest dilution and facilitates the development of well-isolated colonies. Step 10. Incubation. Close the Petri plate, invert it to prevent condensation from dripping onto the agar surface, and incubate the plate at 37 degrees Celsius for 48 to 72 hours. Observation. After incubation, examine the agar plate carefully and observe the distribution of bacterial growth. Typically, heavy confluent growth is observed in area 1, moderate growth in area 2, lighter growth in area 3, and well-isolated discrete colonies in area 4.